1952, Alfred Hershey and Martha Chase at Cold Spring Harbor Laboratory were studying bacteriophage genetics. Phage are viruses that specifically attack and infect bacteria. Phage have an outer protein coat and an inner core of DNA. Phage rely on bacteria to reproduce. They knew from electron micrographs that during infection, phage attach to bacteria by their tails. They assumed that after attaching, genes are pumped into the bacterial host, which then direct the bacterium's enzymes to replicate new phage particles. They set out to determine exactly what caused the transformation of bacteria into a phage-producing factory. Could it be that, as suggested by Avery's work, phage DNA was a transforming principle? From previous chemical analyses, they knew that DNA is high in phosphorus atoms but has no sulfur. Conversely, proteins contain sulfur atoms but have no phosphorus. So, knowing this, they used radioactive phosphorus, 32P, and radioactive sulfur, 35S, to selectively label phage DNA and protein. They then designed an experiment to test which component entered the bacteria for infection. In two parallel experiments, they combined the radio-labeled phage with bacteria that were not labeled. They waited long enough for the phages to attach, and then disrupted the attachment by mixing the culture in a Waring blender. Next, they spun the samples in a centrifuge to separate the phage from the bacteria. Because the bacteria are larger and heavier than phage, the bacteria collect at the bottom of the test tube as a pellet, while the phage stay in suspension in the supernatant. They saw that the 35S label stayed with the suspended phage, and not the pellet of bacteria. The new phage made by these infected bacteria did not contain radioactive sulfur. Therefore, the phage coat, which is made of protein, was not used inside the bacteria to make new phage particles. When they looked at the 32P samples, they found that 32P always pelleted with the bacteria. Moreover, new phage made by these infected bacteria contained radioactive 32P. Therefore, phage DNA was used inside the bacteria to make new phage particles. The phage coat is just the package that delivers the phage DNA into the bacteria. They concluded that the phage DNA alone carries the instructions needed to replicate phages inside the bacteria. So, DNA is the genetic material.